Dear student, I am Dr. Prakash Mungli, Professor of Biochemistry. In this lecture, I will be explaining you Purine Salvage pathways and catabolism of purine nucleotides. Let's get into the details. The purpose of Purine Salvage pathway is to save energy and time, because when a purine ring is synthesized by de novo biosynthesis of purine, it takes about 13 reactions and 7 molecules of ATP to make inosine monophosphate. And from inosine monophosphate to go into adenosine monophosphate or guanosine monophosphate, it takes additional 2 reactions to do that. So that means it takes a lot of energy and time to make a purine ring by de novo biosynthesis. But whereas in purine salvage pathway, it is the readily available purine bases from the diet or from normal cell turnover that is going on in our cells will be used. Purine bases or purine nucleosides are used and simply making a purine nucleotide. That type of pathways are referred as purine salvage pathways. And the greatest advantage of purine salvage pathway is to save energy and time. Now, what are the sources for purine salvage pathway? The source is free purine bases that are coming from the diet or free purine bases that are coming from normal cell turnover. So, what are the enzymes that are required for purine salvage pathway? So, then we need 2 enzymes. One is APRT, other enzyme is HGPRT. APRT is adenine phosphoribosyl transferase, which is used in the conversion of adenine into adenosine monophosphate, whereas HGPRT is used in the conversion of hypoxanthine into inosine monophosphate or guanine into guanosine monophosphate. Let me show you how that happens. So here a free adenine base is available, and this free adenine base will be converted into adenosine monophosphate. You know, adenine is a base. Adenosine monophosphate is a nucleotide. What is the difference here? So, in a nucleotide, we have base, we have sugar, and also we have phosphate. So, what is lacking in the base? That is adenine. It is the sugar and phosphate which is lacking. And that sugar and phosphate, it will come from PRPP. PRPP is a phosphoribosyl pyrophosphate, which will come from ribose 5-phosphate. So, this PRPP is used and ribose phosphate is incorporated into adenine and inorganic phosphate is released. And this is done by adenine phosphoribosyl transferase enzyme, which is simply written as APRT. So, with that, adenine is converted into adenosine monophosphate. In the same way, hypoxanthine is converted into inosine monophosphate by HGPRT. That is Hypoxanthine-Guanine phosphoribosyl transferase. Similarly here, ribose 5-phosphate is transferred from PRPP and hypoxanthine is converted into IMP. So, the enzyme hypoxanthine-Guanine phosphoribosyl transferase, it is converting hypoxanthine into IMP. Similarly, guanine, the base guanine is converted into GMP by HGPRT, same enzyme. So, again PRPP getting into the reaction, going out as PPI. So, like this, purine salvage pathways will make sure readily available adenine, guanine, hypoxanthine, all these molecules will be converted into their nucleotide forms. That is adenosine monophosphate, guanine into guanosine monophosphate, hypoxanthine into inosine monophosphate. And the inosine monophosphate further, it can be converted into adenosine monophosphate or guanosine monophosphate. Alright. So, these are purine salvage pathways. So, that's what I have written here in this slide. And note that PRPP is the sugar phosphate donor in this reaction, and APRT and HGPRT enzymes, they will be inhibited if there is higher concentration of these molecules. Let me show that here in this figure. So, whenever there is increase in the concentration of AMP, this AMP will have a negative effect on APRT. Similarly, whenever there is increase in IMP or GMP, that will have a negative effect on HGPRT. That's the negative modulation that will be going on over purine salvage enzyme. So we have only two enzymes in the purine salvage pathways. One is APRT, other is HGPRT, and it takes only one reaction to convert adenine into adenosine monophosphate, hypoxanthine into inosine monophosphate, and guanine into guanosine monophosphate. Alright, so that's what I have shown here in this figure. Guanine is converted into guanosine monophosphate, while PRPP is a sugar phosphate donor there. Coming to degradation of some of the free purine bases or nucleotides. Basically, there is an interconversion going on in this particular reaction. So, the left half of the pathway I have already explained you. Adenine conversion into adenosine monophosphate, hypoxanthine conversion into inosine monophosphate, and guanine conversion into guanosine monophosphate. Now let's see what happens to the AMP, IMP, GMP molecules when they undergo further degradation. How these interconversion of free bases to nucleosides into nucleotides, that kind of conversion, how that is done in our cells. So as you see here, AMP can go into adenosine formation whenever phosphate is removed. That is done by 5' nucleotidase enzyme. So 5' nucleotidase enzyme, it's going to remove inorganic phosphate from AMP. That's how the nucleoside is made, that is adenosine. Adenosine can undergo deamination process to make inosine, that is done by adenosine deaminase enzyme. And note that if there is any defect or deficiency in adenosine deaminase enzyme, it will lead to rise in the adenosine, and this adenosine, it can also be taken towards adenosine monophosphate done by adenosine kinase enzyme. This is what something we are going to see it in adenosine deaminase enzyme deficiency, which is the cause for 50% of the cases of severe combined immunodeficiency disorder. We are going to talk about that in adenosine deaminase deficiency in detail. Now, when adenosine undergoes a deamination done by adenosine deaminase, we get inosine, and inosine can undergo purine nucleoside phosphorylase reaction to go back into hypoxanthine, and that hypoxanthine again can come back into inosine monophosphate by using HGPRT enzyme, or hypoxanthine can simply go into degradation process. It all depends on the cell, whether it is there in that cell or it is going out of the cell and taken up by other cells. They can use this kind of bases coming from other cells, nucleoside or the free bases coming into the blood, taken up by other cells, and they can use it according to their needs. Similarly, guanosine monophosphate can be converted to guanosine by 5' nucleotidase by releasing inorganic phosphate there, and guanosine further converted into guanine by purine nucleoside phosphorylase, where adenosine, that is ribose 1 phosphate, will be released by purine nucleoside phosphorylase enzyme. So this type of interconversion can go on between free bases, nucleosides and nucleotides in the cell. Now coming to catabolism of purine ring, that is the degradation of purine ring. So purine ring cannot be broken down. That's why it has to be converted into other form, that is the uric acid formation. Note that the degradation product of purine nucleotides, that is adenine in the form of adenosine monophosphate, guanosine monophosphate, so they all degraded into uric acid. So adenine and guanine is degraded into uric acid. So whenever there is higher adenine and guanine available because of increased cell turnover and all that, there is more uric acid being formed. So there are certain enzymes which are involved in the degradation of purine nucleotides. They are AMP deaminase, 5' nucleotidase, purine nucleoside phosphorylase, guanase enzyme, xanthine oxidase enzyme, and furthermore xanthine oxidase enzyme can be inhibited by allopurinol. Let me explain all this to you using this figure here. Now look at adenosine monophosphate, and we have guanosine monophosphate. These are the two purine nucleotides. So adenosine monophosphate undergoes the deamination process and making inosine monophosphate. that is done by AMP deaminase. Inosine monophosphate releases inorganic phosphate and become inosine, which is a nucleoside. This is done by 5' nucleotidase enzyme. Now the inosine will be converted into hypoxanthine upon release of ribose 1-phosphate. So nucleoside is converted to base here. That is done by purine nucleoside phosphorylase enzyme. Now the hypoxanthine is going into xanthine formation, that is done by xanthine oxidase enzyme. And this xanthine oxidase enzyme, it is there in the peroxisome, and peroxisomes are the ones that will lead to purine degradation process. So hypoxanthine conversion into xanthine will give, or it will release hydrogen peroxide as the byproduct, and this hydrogen peroxide here, it will be neutralized into water molecule done by catalase enzyme. Remember this, and that's why purine degradation is going on in the peroxisome, because peroxisomes, they contain high concentration of catalase enzyme. Now on the other side, GMP is converted to guanosine monophosphate by 5' nucleotidase enzyme. Guanosine monophosphate is converted into guanine done by purine nucleoside phosphorylase enzyme, and guanine is undergoing deamination process by guanase enzyme becoming xanthine, and now xanthine undergoes similar reaction where hypoxanthine conversion into xanthine done by xanthine oxidase. So same xanthine oxidase converts xanthine into uric acid. Again there is a formation of hydrogen peroxide here. Now there is an application here with the drug called allopurinol. So this allopurinol, it is a mechanism based inhibitor. It is also sometimes referred as suicidal inhibitor. So what it does? So allopurinol, it is an inhibitor. It has got a similar structure to hypoxanthine or xanthine. So what xanthine oxidase will do is, xanthine oxidase is going to catalyze allopurinol into oxypurinol. Generally, enzymes don't convert a drug into its other form or active form or something, but this is special here. So because it is similar to hypoxanthine and xanthine, so xanthine oxidase thinks that this is a substrate for xanthine oxidase, so it's going to bind with allopurinol in its active site actually, and then convert allopurinol into oxypurinol. So that is done by xanthine oxidase enzyme itself. So what happens with the oxypurinol now? Now oxypurinol, it's a reactive molecule, so within the active site it will bind with this xanthine oxidase irreversibly. So there will be irreversible binding, and it's not released from the active site of an enzyme, thereby it basically denatures that enzyme, and enzyme becomes totally inactive, and that's the end of the enzyme. So while doing so, by this mechanism, allopurinol do not allow conversion of hypoxanthine into xanthine, leading to elevation of hypoxanthine, and also it doesn't allow conversion of xanthine into uric acid, so it leads to elevation of xanthine. So because of this, what will happen? So this xanthine can just get out of the cell, and it will get into alternate metabolic fate and excreted in the urine. And also hypoxanthine, what it does is, hypoxanthine, it will be converted into IMP, inosine monophosphate, by PRPP, using PRPP, which is releasing PPI, and that is done by HGPRT enzyme, and thereby you get IMP. So hypoxanthine is converted to IMP by using PRPP, and then what happens to IMP? IMP will go into AMP formation, or IMP will go into GMP formation, and there will be increase in AMP and GMPs. And this AMP and GMP, so IMP is converted into AMP and GMP also. So this AMP and GMPs further, whenever they are increased, so this AMP and GMPs, they will have a negative effect on the rate limiting enzyme in purine nucleotide biosynthesis in the de novo pathway, and that is PRPP glutamyl amidotransferase. PRPP glutamyl amidotransferase is the rate limiting enzyme in purine nucleotide biosynthesis. So that rate limiting enzyme is inhibited by allopurinol indirectly. So that means allopurinol, it has both the direct mechanism and indirect mechanism to decrease uric acid levels in the blood. What is the direct mechanism? Allopurinol directly act on xanthine oxidase enzyme and inhibit xanthine oxidase, and allopurinol by diverting the elevated hypoxanthine towards HGPRT reaction making IMP and thereby increasing AMP and GMP, and those AMP and GMPs will have a inhibitory effect or negative effect on PRPP glutamyl aminotransferase, which is the most regulated and rate limiting enzyme in de novo purine biosynthesis. So overall it decrease in de novo purine synthesis. So this is how overall purines are decreased, thereby uric acid levels are decreased. So this is what is the purine salvage pathway and degradation of purine nucleotides. And also note that uric acid has got a pK of 5.4, and if the urine pH decreases, uric acid will undergo protonation, and that will crystallize, making uric acid crystals, and that may lead to orange sand appearance in the urine, or uric acid crystals in the urine, or uric acid stones can make if the urine becomes acidic, because pK of uric acid is 5.4, and if the urine pH goes less than 6 or even less than 5, that may lead to precipitation of uric acid as urate crystal, usually as monosodium urate crystal, MSU. Mono means one, monosodium urate crystal. Okay, so this is all about purine salvage pathway and catabolism of purine nucleotides into uric acid. So we have few important enzymes. One is APRT, other is HGPRT, and the third is xanthine oxidase, and allopurinol is the drug that we have discussed in this lecture. Thank you.